Good morning, everybody. Welcome to the MPG Primer. You probably missed us over the last 2 weeks. We're back. We have Kirk Smith, who is thankfully presenting online. He's saving us all from some flu germs. So thank you, Kirk, for doing this remotely. Let's see, Kirk graduated from Virginia Tech in 2015 with a Bachelor of Science in Mechanical Engineering. And then after working as a mechanical engineer for several years, he pursued graduate studies at the University of Michigan, go Blue, where he received a Master's of Science in Medical Engineering in 2019. In 2021, Kirk joined Dr. Udler's lab, where he focuses on using genetic clustering to study complex genetic diseases. I just talked to him, and he actually ran this on one of my favorite phenotypes, nephrotic syndrome. And this is what he's going to talk about today. If you have questions, he obviously might not see you. So just come up to these microphones and cut in at the appropriate time, or raise your hand and I could bring you a microphone. But yeah, let's have some conversations, and thank you, Kirk.
Thank you for the intro, and thanks everyone for joining. And yeah, for those in person, sorry I couldn't be there. As was mentioned, I'm recovering from being a little sick all week, so thought I'd be cautious and present virtually. Um, so yeah, today I'll be talking to you about our lab's research regarding the clustering of genetic loci to study complex diseases. In today's presentation, I'll start with our motivation for performing soft clustering analysis on genetic loci. I'll then describe the actual soft clustering method that we're using, which is called Bayesian non-negative matrix factorization, or BNMF, and we'll go over some of its primary benefits. And then lastly, I'll go through a number of potential applications and downstream analyses that can be done with these clusters. And before I move on, I just wanted to mention that I'll be referencing type 2 diabetes a lot in these slides, since that's the primary focus of our lab's research. However, pretty much everything that we're going over today is applicable to any complex disease. So just keep that in mind. All right, so when most people think of genetic diseases, they think of a monogenic disease such as cystic fibrosis, where a single dysfunctional gene is the primary issue. And we can picture this as a single pipe rupture, which produces a massive but uniform puddle. And then by contrast, complex diseases such as type 2 diabetes arise from dozens or hundreds of variants in different biological pathways. So each variant is only a small leak on its own, but together they pool into a shared disease risk. And because these leaks are coming from various different piping systems, our puddle can be very heterogeneous. And so this metaphor demonstrates why monogenic diseases are, relatively speaking, easier to diagnose and find drug targets for, while complex genetic diseases can be much more difficult in that same regard. So this same concept was captured in the palette model proposed by Mark McCarthy in 2017 to illustrate T2D heterogeneity. On the far left, we have several pathways associated with type 2 diabetes, such as beta cell function, insulin action, and adipose distribution. Then to the right of that, we have patients A through D, which each have a varying degree of dysregulation in these pathways. The amount each pathway is contributing to their overall disease risk will determine their final location on the palette or their final color shade. So this also means that each patient might show different symptoms in the clinical setting or respond better to certain medications. So until we start to untangle this heterogeneity, it'll be very difficult to make strides in personalized medicine. So the question is, how do we begin to do that? There are a number of different approaches that one can use for studying disease heterogeneity, but each has its own challenges. The most straightforward method might be to perform phenotypic clustering, where patients are grouped together based on the traits they have in common, such as BMI or fasting glucose. The issue here is that this clustering is looking at a single snapshot in time. A patient's phenotype will vary depending on where they are in disease progression or treatment. So this means that a patient might change clusters after a few weeks on medication, or had they come into the clinic at a different time point, they could have been grouped into a completely different cluster. Additionally, with this approach, we don't learn much about the disease-causing mechanisms. So that brings us to genetics, which is a much more constant data type, and we can assume it has a one-way causality with the disease. Now, the first step in connecting genetics to a disease is often doing variant discovery using genome-wide association studies, or GWAS. GWAS involve looking at the genetic data of thousands of people and finding variants associated with your phenotype of interest. Going back to our piping metaphor, a GWAS can help us locate all of those tiny leaks, but it doesn't tell us how they're contributing to the disease risk. For one thing, the majority of variants are found in non-coding regions, so we may not even know what gene a variant is affecting. This means that after conducting a GWAS, we still need to figure out which genetic loci are acting together in distinct mechanistic modules. So to do this, our lab's approach is to leverage genetics informed by physiology. So rather than clustering patients based on their genetics, we clustered the SNPs themselves based on their trait signatures. This starts by extracting the T2D associated variants from one or more GWAS, ideally using the largest publicly available studies. Next, we look up those SNPs in GWAS of key metabolic traits such as BMI, glycemic measures, lipids, etc., to get their Z-scores. Finally, we align everything to the T2D risk increasing alleles and put it all together in this variant by trait matrix. This matrix encapsulates how each genetic variant affects each trait, and at this point, it's ready to be thrown into some type of clustering algorithm. What you're seeing here on this slide is essentially a very high-level overview of our entire workflow, and it shows you how we go from variants associated to the disease of interest to looking at the traits that those variants are associated with to putting that information into a clustering algorithm. So now the next question is, what clustering approach do we use? There are a ton of different clustering algorithms out there, which can generally be categorized as either hard or soft clustering. The hard clustering label simply means that each data point is assigned to a single cluster. Some common examples of this are k-means and hierarchical clustering. Determining the true number of clusters, which we call K, can be very difficult with some of these hard clustering techniques. There are many different cluster quality metrics that can be looked at to pick the optimal K, but anyone who has done clustering before knows that often these metrics will point to different values, and the user ends up being required to make some type of gut decision on what metric to use and what final K value to use. So in addition to that, after the clustering is done, the user is also required to determine which traits are driving each cluster. So this could involve doing some type of post hoc trait association testing or some other type of analysis. But essentially the takeaway is that what your clusters are forming around isn't apparent right away when you use most of these hard clustering techniques. Soft clustering, on the other hand, uses weighted assignments. This means that variants can belong to multiple clusters, which is more in line with how biological systems overlap. This is demonstrated in the panel on the right here by the stars, which you can see are in the kind of overlapping regions of these different clusters. Clearly, these data points are kind of in between the clusters, so it's can be expected and that these data points are kind of contributing to multiple signals. So from here on out, I'll be focusing on our specific soft clustering method, which is called Bayesian non-negative matrix factorization, or BNMF. Now with BNMF, the optimal clusters are given as an output of the model. So that takes away any or most of the user bias in terms of picking the optimal K. And then in addition to that, we get the trait loadings for each cluster as an output of the model. So that gives us immediate insight into what is driving each cluster. All right, so I'll now give a relatively quick overview on how BNMF actually works, and I'll mention a few more specific benefits of this method. So to start, the user does have to input a maximum number of clusters to consider, which we'll call Kmax. So typically, this would just be a number that would be way above the number of clusters you expect. So you would just kind of pick a safe K value. So perhaps you think there might be 10 clusters in your data. You could input a Kmax of 20, and then the BNMF algorithm will consider anything below 20. So we then initialize two random matrices, which are called W and H. W will contain the variant weights, so its dimensions are the number of SNPs by Kmax, and H will contain the trait loadings. Its dimensions are actually Kmax by the number of traits times 2. Now, this is because of the non-negative requirement of BNMF. We're actually forced to split each trait into a positive and negative column and then multiply the negative column by -1. So you can see this reflected in the H matrix on the right, how each trait now has a positive and negative, such as BMI plus and BMI negative. So once we have these random matrices W and H, we begin alternating fitting and pruning. The fitting involves minimizing the error between our original matrix X and the matrix product of W times H. There's also a complexity penalty involved to avoid overfitting. Now, the pruning is a very important part because it shrinks clusters that are not contributing to the approximation of X. I've tried to highlight that in this example with the color coding. So you can see how the first column of W is tied to the first row of H and so on. So in this example, we started with a Kmax of 5. However, all of the elements of the fifth cluster were pruned to 0, so we ended up with just 4 clusters. So this is kind of a simple example to show how we go from Kmax to the optimal number of clusters. The BNMF pruning essentially gets rid of all of the unimportant factors. So we end up with the true number of clusters in our data set rather than the K that we started at. Now, this shrinkage is encouraged by the gamma priors, and it also makes our final weights quite sparse. So in an actual clustering run, you would see a lot of zeros or very, very small weights in the final matrices that you get. And so this makes interpretation much easier because you don't have to look at weights, weights for hundreds of variants and traits for each cluster. So how do we define the clusters once we get these final W and H matrices from BNMF? Yep.
Oh, sorry, I was muted. Can I ask a quick question? Is there an
Yes.
upper bound on the number of traits you could put in? Because I'd imagine with type 2 diabetes, you have some ideas of where to look. But if you're unsure what's driving your phenotype, you might want to include a lot more GWAS traits. And I don't know if there's an upper bound to the number that you could put in.
Yeah. Okay. So I have in 2 slides. I'll go over that.
Oh,
I
great.
think it's 2
Good setup.
slides. Yes. Maybe 3 slides, 2 or 3 slides. I'll cover that. So I'll hold off on answering till then. Um, Okay, so in terms of defining the final clusters, one approach would be to consider all SNPs and weights with a non-zero weight, or sorry, all SNPs and traits with a non-zero weight. There's nothing wrong with that approach. However, our lab's approach has been to focus more on the components that are most heavily driving each cluster. So to do this, we compute a cluster specific, or sorry, we compute a weight threshold to maximize signal to noise ratio. So from there, all of the SNPs and traits with weights above that cutoff are assigned to a cluster. I've demonstrated that here by highlighting in yellow the variants and traits that are above the cutoff in each cluster for this example. So then if we wanted to zoom in on cluster 1, for example, we would see that SNPs 1 through 4 are above the cutoff. So those are included in the cluster. And then we can see right away that we also have increased BMI and glucose in this cluster and decreased height. So you can see right away how having these trait loadings makes it super easy to begin interpreting these clusters immediately rather than having to do some post hoc testing. And then for anyone that ends up reading any of our papers, you might see, you'll see these circle plots, which are sometimes we get asked about, but these are just essentially showing the same information as the bar plus to the left, just showing um, the weights of the variants and traits that are driving each cluster. So same information that I just described here, just in a kind of nicer format. So a few more quick notes on BNMF. Certain inputs and traits might not end up in any cluster, which you might have noticed on the last slide. So this will depend on several things, such as the BNMF pruning or whatever approach you take for the weight cutoff. With our method, we often see around 50% of SNPs contributing to one or more clusters. And in line with that same idea, one kind of nice benefit of this method is we're not forcing SNPs into clusters that they don't belong. So say there's a SNP associated with your disease that doesn't end up in any of the clusters. This, maybe is disappointing at first, but in reality, it could be very informative because it tells you that that SNP is not acting through a mechanism that's captured by the traits you're using. So looking at this example in subpanel 2, if we were using a method like k-means, we essentially would have had to stretch some of these clusters to encapsulate these outlier data points. So not only would we kind of be maybe wrongly classifying those data points, but we could be skewing the cluster centroids, which are later used for some other downstream analyses. And then lastly, it's important to remember that your results will depend on the quality and quantity of your genetic inputs. So the more powerful GWAS that you have means the more SNPs you have as inputs, and also the more reliable your variant trait associations will be. And then additionally, the number of traits you input will have a big impact on the number of clusters that you get. However, a big caveat there is that it's not necessarily the number of traits, but the number of relevant traits. So now getting kind of into that trait selection question, our group's approach has been to take a data-driven approach for deciding on what traits to include. And this is probably actually the most common question that we get for running BNMF is how do we go about deciding on what traits to include in this variant trait matrix? So there is no correct answer for this. Some people prefer to handpick traits, but our method has been to include as many trait GWAS as feasible. And the thing to keep in mind there is that that involves downloading and formatting all of these trait GWAS, which is actually probably the most time-consuming part of this entire workflow. And also, the more traits that you include in the pipeline, the longer it's going to take to run. So it's just something to keep in mind. It's a limitation that can be overcome, but it's to give you an idea. In our most recent paper, I think we included about 160 traits initially, and then the final matrix ended up at around 110 traits. And then so this slide kind of goes over how we get from that 160 to that 110. So once the initial variant trait matrix is formed, we have our T2D SNPs on the y-axis, and we've pulled all the z-scores for these initial traits that are on the x-axis. Now we start pruning the traits. So the first thing we do is we remove traits that have low sample sizes. This sounds like something that could be done prior to running the pipeline. However, often GWAS studies will report a maximum N. So they might say that they took the genetic data for up to 15,000 people. However, once you start actually pulling SNPs and data from the summary statistics file, you might find that a lot of the variants that you're interested in, they actually only measured in a few hundred. And we typically look for a median sample size of at least 10,000. So sometimes we do lose traits due to a lot of the SNPs not having enough sample size. So next we remove traits without at least one Bonferroni significant SNP. This is probably the most important filter because this is kind of what allows us to throw traits into the pipeline that maybe we suspect are associated with our disease, but we aren't entirely sure. And then if it turns out that none of our T2D SNPs are associated with that trait, we simply remove the trait and therefore we're not adding any unnecessary noise to the clustering. And then finally, if we have two traits that are highly correlated, we just keep the one with the strongest signal. This is to make sure that the traits are representing independent signals when the clustering is done. And kind of a common example for this is we often will include two versions of a trait, one adjusted for BMI and one unadjusted, and then we can let the pipeline figure out which one to keep. So hopefully that answers the question about kind of picking traits to include. Now, as I mentioned, this whole process can be very, there's a lot of steps to it, from formatting the GWAS, pulling the summary statistics and everything. And for the original paper that Dr. Udler published on this topic in 2018, all of this summary statistics wrangling had to be done by hand, which I can't imagine. Fortunately, right before I joined the lab in 2021, a few members of our team created this high throughput R pipeline, which automates almost the entire process. Essentially, the only requirement from the user is a list of file paths for the input GWAS. So at this point, pretty much the main work for someone that wanted to start a new project would be to figure out what traits they want to include. And if we don't have those trait GWAS already available somewhere on our server, they would have to download and do some formatting to the summary statistics files. And then they input those paths to the pipeline, and pretty much everything else runs on its own. So this means that we can now apply our method very efficiently to any complex disease of interest. As you can see in this list, we've already published several papers on diseases and traits outside of type 2 diabetes, and we have several more in the works. All right, so I'll now get into applications of the genetic clusters. So probably the most useful downstream analysis we can do with these clusters is generating cluster-specific partition polygenic risk scores, or cluster PPS. The steps for doing this are very similar to what would be done for a full disease risk score, such as a T2D PRS. As we discussed earlier, we start by defining each cluster's variant set, shown here with the blue bars for cluster 1. So this includes any SNP that was above the weight cutoff for that cluster. For a given patient, we then pull their genotype dosages at those SNPs. These are shown in the blue circles, and so these will be either a 0, 1, or 2, depending how many alleles that patient has for the given SNP. We then compute the dot product to get a partition PRS for the cluster. This is repeated for each cluster to build a PPS profile for that patient. We can then determine each patient's dominant genetic pathways and compare PPS profiles across patients. So as you can see, looking at this example of patient 1, we can see that much more of their T2D risk is coming from cluster 1 than from cluster 12. And this is tied to their allele dosages for the SNPs in each of those clusters. Sorry. All right. So once we have cluster PPS generated for a cohort, we want to see how those clusters are associated with various phenotypes. So clinical data can be, or is typically very noisy. So these patterns are often not visible at the individual level or from a simple correlation test. However, once we employ more powerful regression models and account for covariates such as age, sex, and race, we start seeing some really nice signals for these clusters. In cases where we are testing traits that we used in the clustering, we typically think of these as validation tests as the clusters. So for example, confirm, as you can see here in the kind of the top plot, confirming that the obesity cluster has a strong association with BMI. Now, when it's a new continuous trait, we consider an exploratory test. Now, we can, of course, also do the same analysis with discrete outcomes, such as shown in figure 2. So we can look at how cluster PPS are associated with other diseases such as CAD or CKD. And in the same manner, we can account for things such as age, sex, and race so that we're only looking at the effect of the cluster on that outcome. And then while I won't go into detail on this today, I wanted to note that we're also able to generate cluster PPS at the GWAS level. And this is can be a really helpful analysis because that means we can run this for traits that aren't available at the individual level, or perhaps if we want to take advantage of a larger sample size that a GWAS might have over an individual level cohort data set. So it's also of interest to determine which tissues and cell types the clusters are active in. To do this, in our recent paper published in 2024, we created credible sets for the variants in our clusters, and we then overlapped the credible set variants with cell type and tissue type genomic annotations. This was done in these two epigenomic data sets, CATLAS and Roadmap. And then finally, we used permutation testing to determine the cumulative posterior probability for each annotation. The results you see on the right show the significant tissues and cell types for each cluster. And we saw some really cool results here that supported our interpretation of these clusters, such as the beta cell clusters being enriched for pancreatic islet annotations and the liver lipid cluster for hepatoblast annotations. So this is kind of confirming that these genetic clusters are capturing variants with distinct effects in specific tissue and cell types. Now, in addition to studying how the clusters behave across tissues, we can also look at how they differ across populations. In these figures from our 2024 paper, on the left, we see the proportion of total T2T risk coming from each cluster broken down by population. For example, we see in East Asians in blue, the two lipodystrophy clusters account for a notably larger fraction of total PPS than Europeans or Africans. Now, just a quick note, you'll notice that for each of these clusters, the kind of maximum value is very different across each cluster. And that's based off of the total sum of weights for each cluster. And so for this analysis, it's important to note that we're not comparing risk across clusters so much as we are looking at populations within a cluster. If you wanted to somehow try to look at the risk coming from each cluster, you would have to probably normalize the weights or the PPS across clusters before doing that type of analysis. So kind of the takeaway here is that we're looking at population differences within a cluster. So kind of going back to this difference that we see in the East Asian population in the lipodystrophy clusters. In figure 2, we modeled T2D risk versus BMI and plotted these as curves for each ancestry group, and we adjusted for age, sex, and PCs. So you can see that the Europeans in orange have a T2D risk of about 9% at a BMI of 30, which is kind of the standard threshold for obesity, while East Asians have this same risk at a BMI of only 24. Next, we adjusted these models for the lipodystrophy 1 and 2 PPS, and we saw that this difference in risk decreased by about 67%. So this is essentially quantifying how the genetic differences in these adipose dysfunction pathways are explaining part of the well-known lower BMI threshold for T2D in East Asians. So this was actually probably the coolest result from our 2024 paper, since this tendency for East Asians to develop T2D at lower BMI is a well-known phenomena, but it hasn't been studied to a large degree or figured out. So the fact that these clusters are capturing this difference was a really interesting finding. Finally, I wanted to highlight some really exciting work from Dr. Melina Klausnitzer's lab. So from subcutaneous and visceral fat biopsies, Dr. Klausnitzer's team derived mesenchymal stem cells. The cells were differentiated over 14 days into mature adipocytes with imaging and molecular profiling at days 0, 3, 8, and 14. Now, when they stratified by total T2D-PRS, there was really no difference in lipid accumulation or mitochondrial markers. However, once they stratified by high versus low lipodystrophy PPS, there was a huge signal in the subcutaneous day 14 adipocytes. This included differences in lipid droplet size and mitochondrial activity. So this analysis was really cool in that it demonstrated how these genetic clusters are actually capturing functional differences in cellular biology. Now, before wrapping up, I wanted to mention that these genetic clusters are constantly evolving as GWAS and computational methods become more powerful. As I mentioned earlier, Dr. Adler first published this work in 2018, and then the development of our high throughput pipeline allowed us to expand that analysis in 22. So that included increasing our number of SNPs by over threefold, in addition to adding more traits. And then more recently in 2024, we published a new set of clusters that expanded from European-only GWAS to multi-ancestry. So in addition to GWAS becoming more powerful and diverse, we're also constantly working on ways to improve our BNMF pipeline. And I just wanted to kind of mention this because it goes to show that even if genetic clustering has been done on a disease or phenotype that you're interested in, there's almost always room for improvement. And kind of in line with that, for anyone that's interested in applying our workflow to their own research, we do have a GitHub page that, as I mentioned, we are constantly trying to update it and make improvements to our workflow. So you're welcome to check this out and try running the clustering yourself. Or if that seems a bit overwhelming to you, feel free to reach out to Miriam or I if you have any ideas for a collaboration project. And then lastly, I wanted to touch on the limitations. I know I've kind of painted this approach as some magic clustering tool today that's perfect, but as with any clustering method, there are some limitations involved. So number one, the trait inclusion, as I touched on this a bit earlier, but we're still at the stage where we have to manually download and format each trait GWAS before we can include it in the pipeline. So this is actually, again, as I mentioned, probably the most time-consuming part of the workflow. For example, the UK Biobank has several thousand brain imaging-related traits, and it would be great if we could throw them all into the pipeline for our Alzheimer's project and see what sticks. But the data processing is a lot of work, and the more traits you include, the pipeline can also take exponentially longer to run. And then another one also kind of relate to the trait inclusion is data availability. I think this mostly applies to projects focused on non-European populations. While the population you're interested in might have a powerful enough GWAS for the main disease you want to study, if there aren't enough trait GWAS for that population, then you might be a bit stuck. You can always fall back to using multi-ancestry GWAS, but this is just kind of a limitation to consider. And then finally, something that we're looking into a bit more now is tuning the hyperparameters of the BNMF algorithm. There are things you can adjust, such as the tolerance threshold or the gamma prior, that will affect the clusters that you end up getting. So I think for future papers, it might be important to do some type of sensitivity analysis on these hyperparameters. And I think this could actually be a really useful analysis because it might help us identify the most robust clusters. So in conclusion, complex genetic diseases lead to heterogeneous phenotypes. Soft clustering of genetic loci by associated traits using BNMF is a unique approach for untangling this heterogeneity. And then variant trait groupings provide mechanistic pathway insights, while cluster PPS are useful for patient stratification. And kind of takeaway for anyone interested in applying this to other diseases is that this method is very flexible, and we can apply it to essentially any complex genetic disease as long as the data is available. And lastly, I just wanted to say thank you to everyone in my lab and Dr. Udler's group, and also to the many other collaborators that have helped on this project over the last few years. Yeah, and that's it.
Let's thank Kirk again. And next 2 weeks we'll be in here. So see you soon, and please join us across the hall for Steve Parker, also from U of M.